Hello, my name is Nikolina Radulovich, and I am a staff scientist at University Health Network in Toronto, Canada. And for the past 5 years, I have been leading a next-generation biobanking facility, Princess Margaret Living Biobank, and my experience lies in the generation of patient-derived organoid models. And today I would like to give you an overview of what we have accomplished and go over some lessons we have learned along the way. So this is an overview of presentation. First, I'll give you an overview of Princess Margaret Living Biobank, who we are, and lessons learned from establishment and propagation of tumor organoids, and also to give you some or show you some lessons that we learned from drug screening of tumor organoids, and tell you a little bit about current and future directions of the core. So as many of you have probably used biobanks in the past, biobanks are biorepositories that accept processes and stores and distributes biospecimen and also associated data for use in research and clinical care. Now, historically, biobanks have been depositing mostly non-live tissue that had limited applications. Living biobanks, on the other hand, involve biobanking of live tissues, either through cryopreservation of tissues for later use, or generation of patient-derived models that could be cryopreserved and renewed. So these living tissues can then be applied in preclinical testing, biomarker discovery, toxicity evaluation of pharmaceutical agents, and also it can be used to drive precision medicine approaches in treatment of diseases. So there's three types of patient-derived models that have been used for biobanking and subsequently for research purposes. These are established cell lines from the patient material, patient-derived xenograft models that are grown in mice, and also patient-derived organoids. Now, currently, organoid models are gaining a lot of traction in this field because they closely mimic phenotype and genotype of original tissues compared to cell lines, for example. They're cheaper and faster to grow than PDX models. They grow in large numbers, so they're amenable to large drug screening efforts. Some disadvantages include the fact that these models are void of any cells from tumor microenvironment, and we're still not sure how their behavior mimics the clinical behavior, and also they're quite expensive to grow. Now, the Princess Margaret Living Biobank has been around for the last 5 years, and we established it in hopes to consolidate existing patient-derived models and also to make new patient-derived models for effective distributions of resources to research community. We also wanted to optimize model sharing of models and data for our institute, but also for other academics and different companies. So the way it works that we receive tissues or fluids from biopsies or surgeries of Princess Margaret cancer patients that are diagnosed with various types, and you can see the types that we are currently working with in this figure. We generate the patient-derived models and then biobank them for future use. There are two arms of the facility. One arm, led by Dr. Nguyen Pham, generates and biobanks xenograft models, while the other arm, that's led by me, develops technology and generates organoid models and biobanks them for future use. So the focus of this talk will be on the organoid core, but I do encourage you to check out more of the collection of the xenografts, and you can also go on our website too for more information. So who we are? So we are, again, the Living Biobank Organoid Core, and the way we generated this core is we really collaborated with our researchers and the UHN Biospecimen Biobank that allows generate specific consenting processes and protocols to generate these models. And we are the first UHN next generation biobanking facility. We established a central repository of patient-derived organoid models, develop SOC and Q&A for PDO generation and use, And also we provide these materials and services and models for the use of PDOs to our own researchers, but also external academics. We support our researchers by providing these models, and also we try to enhance the current quality of publications and maximize the grant awards. So most of you are probably aware what organoids are. I'm going to quickly just remind you of what this technology is all about. So this was pioneered by Dr. Hans Cleaver's group, and it's based on 3D growth of tumor or normal cells derived from adult tissues. They can also be derived from embryonic tissues or iPS cells. So this technology has been developed over the last decade, and the protocols have been established for generation of organoids from different organ sites, and some of these are summarized here. Now, the technology involves the association of cancer or normal tissue from patients and subsequent plating of these dissociated cells in extracellular matrix, such as nitrogen media containing tissue-specific niche factors that support the proliferation and differentiation of organoids. And of course, the different niche factors are used to grow different types of tissues. And you can also grow both normal and cancer organoids using this technique. Over the years, this technology allowed for generation and subsequent biobanking of patient-derived cultures, and uses of these models range from cancer modeling, gene profiling, drug efficiency testing, and regeneration medicine. Now, the success rate of organoid generation varies across different tumor types, as well as across specimen sources used to generate these models. And here are some numbers that have been reported in the most recent review article by Cleaver's group. So first, you can see that efficiency varies from 100% to 15%. However, one of the issues that we have run into as a biobank is that when actually looking at this data and applying it to our success rates is how the term efficiency is actually defined. So efficiency is still defined differently across different publications and groups. Some groups define efficiency as initiation rate, or whether or not it's a yes or no growth of organoids, while others define them as models that grow a particular amount of time in the culture. In addition, the quality assessment in these publications is not standardized, and all of this might not be an issue for a research group, but is definitely an issue for Living Biobank, because we want to make sure that the models that we have in the biobank are renewable, they can grow for long periods of time, and that they have predictable growth patterns. So we have quickly learned that reaching some of these published organoid generation efficiencies have been quite challenging, and for the most part not met. So while we start with already established technologies, we have optimized many protocols in the house. So I will a little bit about our workflow. So basically, when we get the tissue from the patients that are consented, we record this in our tissue database. We de-identify the patient, and we always keep a little bit of a tissue, the snap freeze the tissue, and also embed it for histology. The tissue is then processed in our facility, where we dissociate the tissue, embed the cells in the matrigel, and we establish the optimal growth conditions for that particular model. Our goal is to establish long-term cultures that we define as models that grow for at least 6 to 10 passages, and they have a predictable splitting ratio, and they can be renewable. So once we know that the organoid is going to make it as a long-term culture, we then do extensive characterization that includes genotype matching to the original tissue, histology characterization, which includes using different immunohistochemistry markers that are used to diagnose the specific disease. We perform mycoplasma testing, doubling rate, and also flow analysis is sometimes important, particularly when making these models from xenografts, and I'm going to get into that in a bit. And then we generally biobank about 10 cryovials per model, and this is under the patient-parent distributed sample, and a lot of these organoids also come with some clinical data. And then we document SOPs for each of these models. So now I would like to get into some of our collection, just to show you have a brief overview of what these models look like. So the colorectal organoid models, we have generated about 46 long-term models. Success rate varies between 75 to 80%, with 80% success rate of biobanked models being the models that are generated from xenograft tissues. We have a variety of models from either primary resected metastatic tissue, from xenograft tissue, or from the normal tissues. The maximum passage ratios for these models is 1 to 16, and the minimum is 1 to 2. And the medium splitting ratio is about 1 to 7. And these are some histology representatives. So the histology varies depending on if it's a normal or tumor organoid, of course, but the tumor organoids are usually small-sized gland-forming epithelial clusters with either large or small lumens, and sometimes they have multiple lumens as well. Most of these are CK20 positive, and this is not shown here. Our next set of, and I would say our biggest collection, is from organoids that were generated from patients with pancreatic adenocarcinoma, or PDAC. We have generated models from both surgical tissues, from the biopsy or metastatic tissue sites, and from xenograft tissue. Our success rate is from 50 to 80%, with 50% being success rate of biobank samples generated from biopsies, and 80% from xenografts. And challenging was to actually make the models from biopsy, simply because of the small amount of material that one really gets from biopsies. They grow quite fast. Usually the average splitting ratio is 1 to 4 every 11 days, and this is the typical histology. Again, these are mostly clusters of cells with thick epithelium and varied lumens. They have moderate to high nuclear pleomorphism. They have some mitotic events, and also they're all CK19 positive. Our next set were lung organoids, and we have actually published a paper on this. They have been challenging to grow, to say the least. We have been growing them for non-small cell carcinoma patients and also from small cell carcinoma patients. Success rate varies from 10 to 50%. Our best success rate is growing them from small cell lung cancer tissues and from non-small cell xenograft tissues. The biggest issues in growing these from the primary resected tissues is the normal outgrowth that has been documented in literature, and that definitely has been one of the issues in the core to generate these models. But the passage rate, the average passage splitting ratio is 1 to 3, and usually every 10 days to 14 days. And here is also we have a doubling rate for these models. Generally, the doubling rate is about 58 hours. Histology varies with the diagnosis. So adenocarcinoma, squamous, small cell carcinoma, they all have sort of different histologies. For adenocarcinoma, organoid models are generally TTF1 positive, where squamous shows positivity for P63. And these are some of the representative histologies. Another set of models, esophageal organoids from esophageal adenocarcinoma. We've grown them from both resected tissues and biopsy tissues. The success rate is from 50 to 70%. These models grow pretty fast, 1 to 4 splitting ratio every 11 days. And the doubling rate is about 54. Average doubling rate is about 54 hours. This is what some of these organoid models look like. They're clusters of cells, and they have varied lumens. So now the last two tumor types have been very challenging. So one of them was ovarian. While majority of our ovarian tissues will generate or initiate organoids, not many of them can actually be biobanked because they grow only for a short period of time, which sort of indicates that the media is not, or the technology is, not adequate to grow these models. But the average, and also they grow a bit slower than the rest. So the average splitting ratio is about 1:3, and they're generally passaged every 2 to 3 weeks. And the doubling rate is also about average 72 hours. So our highest success rate would probably be from ascites fluid, and of course, our lowest success rate would be from the primary resected cases. Biopsies are super difficult. We haven't really been able to grow too many long-term organoid models from those. And finally, breast organoid models. Again, we have processed many tissues and you use many different types of medias. And however, we have generated organoids that can be biobanked. Most of these organoids, so the success rate is low, is 10 to 30%. And most of the ones that we actually made are either high grade or triple negative. So the low grade are generally very difficult to make. And number one issue, again, if I was to show you initiation rates, it would probably be anywhere between 90 to 100%. Most of these will grow something. However, the major issue here is the contamination of normal cells and of the normal organoids. So this is an important thing to address moving forward, because it's important to incorporate appropriate QCs when using these models in early passages, because if you want to use them for drug screens, you got to make sure that whatever you're growing is not of normal origin. So that is sort of our collection, and I included here 2 slides to show you some of these issues that we've faced, particularly on normal outgrowth on breast and lung, when using breast and lung surgical tissues. So these normal organoids have certain characteristics that distinguish them from tumor organoids. For example, in case of breast, they're basal and show CK5 positivity on the edges of the organoids. In the case of the lung, they show positivity for P63 even when grown from adenocarcinoma specimen, which are not supposed to be positive for those markers. The organoids are very uniform in size, and there is show no nuclei atypia. In addition, based on our observations, these models are short-lived. They can only grow for up to 10 passages. They're hard to revive after freezing. It usually, what we do, if histology hints that these models could be normal, we then use SNP analysis to identify the origin of the organoid. For this, we send the tumor, normal tissue, and the organoid from the same patient, and then we perform the SNP analysis. So here's an example of 2. So the first one turned out to be a tumor organoid. So this is all refers to the CNV. This is the CNV profile of the organoid. It matches to the CNV profile of the tumor. Whereas the other model, organoid model, you can clearly see the CNV changes in the tumor that are and completely silent genotype genome in the normal organoids. So again, very important to utilize QC methods in your research if you're using models, organoid models for breast and lung, because especially if you're drug screening them, you want to make sure that what you're growing are the tumor organoids. Another thing that we have encountered, or that it's when we grow organoids from xenograft tissues, sometimes we'll get mouse organoid contamination, and they kind of look quite interesting. So here is an example of a breast organoid that's derived from xenograft tissue. So as you can see here, there seem to be two populations of organoids that are apparent here. Small organoids and really large dark organoids. So these large dark structures are actually mouse cells. And if you're not properly handling them, they will take over the entire cultures. And you might end up working with mouse cells without knowing. So we have incorporated a QC step when processing xenograft tissue. So we use H2K antibody depletion. So this way we deplete mouse cells after dissociation. So in hoping to enrich for the tumor cells that are actually being plated for organoid generation. While this has improved our success rate, there are still many cultures that have a large proportion of mouse organoids that just like to grow. And so if working with such cultures, one should periodically perform flow analysis to confirm that you are indeed working with human tumor organoids and not mouse. So again, if using these for drug screens, this QC step seems to be necessary. So these are the lessons that we have learned. Basically, we need to establish the clear criteria for organoid establishment definitions. What is initiation? What are short-term? What are long-term cultures? We try to address the normal outgrowths and perform proper QC to identify it. Insufficient starting material is always a problem. If you don't have enough tumor cells, it's very difficult to initiate these organoids. Mouse contamination should be addressed, and continuously improve the organoid technology, whether or not it's by using multiple medias, because we need to consider heterogeneity of these tissues, using alternative to matrigel, and also find ways to eliminate the normal outgrowth. So let's now look at the ways we utilize these models for different projects. And we work with many principal investigators at UHN, external academics, and for-profit companies on projects that utilize these models. For instance, for drug screening efforts. What I will highlight here are some results from already published papers. So one of the first papers we published was with Dr. Ming Tsao's group at UHN. So we published a paper on generating organoids from non-small cell lung cancer tissues. So our technology had sort of a varying success rate from 0% for primary squamous tissues to about 30% from lung adenocarcinoma xenograft tissues. We have also utilized some short-term cultures for drug screens. And in one of our experiments, we assessed the correlation of drug response between organoid and parallel xenograft models. So this is the second, this is the set of graphs that you can see here. So basically, the organoid screen revealed sensitivity of KRAS mutant models to MEK inhibitor trametinib compared to KRAS wild-type models. So the KRAS model is the orange line here. And the same experiment was then performed in vivo, and the results also revealed differential responses of KRAS wild-type and KRAS mutant xenograft models to trametinib. Another paper that we have collaborated with Dr. Gail Darling and Dr. Jonathan Young from UHN is to generate and screen organoid models generated from patients with esophageal adenocarcinoma. So the establishment rates here was about 57.2%, we published the characterization of 5 models. These models were drug tested and characterized. So the primary, if you can look at the histology pictures here, the primary tumor in all 5 endoscopic biopsies showed a spectrum of morphological features ranging from well-formed glands to cribiform nests to sheets of cells, demonstrating this presence of heterogeneity. And overall, the organoids that were derived from these tissues recapitulated the cytological features of these biopsies. Whole exome sequencing was also performed, and it revealed similar mutations of EAC driver genes in organoid tissue pairs. Organoids were then treated with single chemotherapy agent, each of which are part of a clinically utilized induction regimen. And overall, the organoids displayed various sensitivities to different drugs, which kind of suggests an individualized response to each chemotherapy reagent. In this case, we did not have, there was a first paper, it was a small set. We had no corresponding patient data, but currently we're working with the team on a bigger set. And that sort of is the publication that's in the pipeline. And that publication will more directly address the correlation of organoid drug response to the patient response. And another one, this is the most recent paper we have published. So this is on generation of xenograft and organoid models from resected pancreatic, duodenal, and biliary cancers. So we focused on efforts on generating organoids from pancreatic adenocarcinoma xenografts here. And these are some of the representative models. These models are actually rare KRAS wild-type models of pancreatic adenocarcinoma. One of these models was OSIP256, and this was shown to have an oncogenic in-frame BRAF deletion mutation. And current clinical trials are sort of underway to test a possible inhibitor for this BRAF mutation. That's a Ly300-9120 inhibitor. And we actually tested efficacy of this inhibitor in both in vitro and in vivo model of OSIP256 patients or absence of MEK inhibitor trametinib. So you can see here in both of these RAF or in vitro, the Ly300 alone showed minimum efficacy on organoid growth. That was also shown in vivo. However, the combination of Ly and trametinib showed better efficacy in both organoid and xenograft model. So another thing that we have discovered as working on this project is that organoid models can actually be generated from properly cryopreserved tissues. So we had a big biobank of these tissues that were cryopreserved, and majority of the models were actually made from cryopreserved tissues. And again, also this data also says that, you know, if there are many xenografts already available, it's useful to actually generate these organoids from these models because they're better used for the first line discovery and you know, such as larger drug screen panels, and you know, you can always do a confirmation of the response in vivo. And also, majority of the organoid models that we have generated can actually be engrafted, and that's another good test for to assess whether or not the organoid is actually tumorigenic. So those are our results, and the lessons, of course, we learned from these drug screens is that you know, if you're screening the long-term organoid models, you actually get consistent results across technical and experimental replicates and across different passages because you can predict how the model grows, and there's some correlation with in vivo response. The screening of short-term models you have to be careful with these because you have to include a lot of QC steps here. For instance, you might have to enrich for epithelial content by using sorting. You might have to include doubling rate controls to make sure that your organoids are actually doubling at the time during the drug screening period. Also, tumor purity controls are important, such as histology and SNP analysis to make sure that you're not drug testing mouse cells or normal cells. And also, you know, it's also good to culture them for a few passages before drugging if possible at all. So our future directions are in improving our organoid establishment rates across different tumor types. Also, we are working with many teams at UHN to generate complex models, including cancer stroma and immune cells, and also working with translational clinical teams to actually get some more idea of how correlative organoid response is to actual patient drug response. And to sort of finish this off, you know, we have worked with many UHN researchers, but we have also worked with many different academics and different for-profit companies. we can distribute models. We can also work with you on the drug screening projects. We also do quality assessments of existing organoids, and there are many custom projects that are happening in our facility. We have worked with number of collaborators shown here, and our list is growing every day. I said we have been around for 5 years, but really our collection has increased in the last few years. These are the acknowledgements, and I will finish sort of by acknowledging many of our staff from both organoid and PDX cores, and many talented people here. Also our PI collaborators that have been a key in helping us develop technologies to grow these models. We really have them as our advisors, and we have been getting grants with these PIs and publishing along with them. Also, the careful model characterization is something we are particularly proud of in the core, and this would not be possible without many other cores in our institutes, including genomic cores, bioinformatic cores, profiling labs, and UHN biospecimen biobanks. And that's it. Thank you for your attention.